Our final speaker for this afternoon is Dr. Rebecca Roach. Beck is a research scientist with the Department of Agriculture and Fisheries, also based at the Ecosciences Precinct in Brisbane. And Beck will be talking about bacterial diseases of lettuce. And we'll just make sure that we are sharing Beck's screen. There we go. Excellent. Over to you. Thanks, Beck.
Okay, thank you, Heidi. Hi, everyone. Like Heidi said, I'm Rebecca. I work at the Ecosciences Precinct here with QDAF. I'm going to take you through some bacterial diseases of lettuce that you may or may not commonly see around. There we go. So just as an overview, we have a few options, a couple of leaf spotting types and some others that you might find either in the soil or just throughout the plants generally. So we have bacterial leaf spot caused by Xanthomonas tortorum, Patibar vitiens. Previously Excampestris, that one's had a name change fairly recently, I believe. You can also see soft rots. These are actually quite general throughout vegetables, caused by various species in Owinia and Pectobacterium. Those also had a few name changes. Corky root is another one caused by Rhizomonas superfasciens. That one I have encountered once or twice, but less common. And finally, varnish spot, midrib rot, I think sometimes also called tar spot, caused by Pseudomonas chicorée, and that one I've seen a fair bit of. So first of all, bacterial leaf spot. Like most of the bacterial leaf spots in vegetables, it's caused by a Xanthomonas species of varying kinds. Leaf spots in general are encouraged by high humidity and rainfall, and they're pretty generally spread by water, and a lot of them have the potential to be seed-borne as well. This one I don't personally see a lot of. I don't think I've encountered a bacterial leaf spot in lettuce as such. In every brassica under the sun, basically, sure. But yeah, this one, maybe we don't get as much, maybe I just don't see it. I'm not fully sure. The picture you can see here is a first report fairly recently of bacterial leaf spot from Turkey. So next we have the soft rots. Yeah, like I said before, they are found fairly often or fairly widespread throughout vegetables. The Pectobacteriums, for whatever reason, will get in and cause a fairly gross oozy rot like you can see in this picture. Again, higher humidity and rain, really any period of sort of field and leaf wetness will encourage those. These can absolutely be associated with post-harvest disease as well. So maybe something you might not see until the very end, unfortunately. And certainly when it comes to a lot of soft rots, it might not always be one pathogen. It might not always be a pathogen that got in there initially. It could have been some kind of damage, some kind of environmental condition that has done the damage previously and allowed the soft rot bacterium to come in after it and cause the rot. And next up, we have Corky root, Rhizomonas superfasciens. So this one we do have around. The Rhizomonas and associated Agrobacterium type things are quite slow growing organisms. They can be quite difficult to isolate as well. They sometimes require specific methods and isolation techniques to get them out and detect them, as they're a soil dwelling bacterium. They can also be easily confused with sort of native agrobacteriums that do already exist in the soil. So you have to be quite careful when detecting those. The pictures you can see here are a suspect Corky root sample that we got a while back. Again, with the difficulty in isolating, we may not have actually found this one out for sure. It's not one that I see too often, but we are actually in the process of improving our enrichment methods and isolation methods for species like this, which will be really good. So yeah, it's getting a lot easier to detect those basically. There's also some evidence that the soil microbiome will really affect how badly this disease could flare up or how often it might happen. So like obviously heavy chemical usage is going to be killing off most of your good soil microbes. And that could be encouraging, you know, all sorts of root infections that might otherwise not be as much of an issue. Yeah, I think there's certainly been some work on that that suggests that might be the case. Next up, we have varnish spot. So this one I have seen a fair bit of, and we generally get it out of the leaf lesions that you can see here. Apparently, this has been isolated from roots and soil as well. In that case, foliar bacterial diseases don't generally survive in the soil particularly well. It's more about trash, infected trash from the last infested field. But again, with a lot of bacterial diseases, especially the leaf ones, high humidity, rainfall, spread by water is generally how they're encouraged. Ensuring good, you know, plant spacing, especially when they're seedlings and things. A good opportunity to air them out is very important to avoiding foliar diseases. So the pictures you can see here came from a survey we did in 2018 in Lockett Valley. And you could mostly see there's large lesions sort of down the midrib here. And the plants can often look quite all right from the outside, but when you start hacking into them, especially at like early infection stages, you likely wouldn't be able to tell how badly infected they are until it gets bad enough that you start seeing them fully rot. So just to go over a few diseases that might look similar to some of these bacterial diseases, just to help you in figuring out, you know, what might actually be going on. Here's a few nice pictures that will show you how tough some of these can be to work out. So on the left down here, up the top, there's a little lettuce leaf with a couple spots, fairly benign looking. This we're pretty sure is environmental or some kind of, you know, spray damage, something like that. At the point where, you know, bacteriologists, the mycologists, the nematologists all look at it and say nope, nothing. Yeah, it's probably environmental, but it looks quite similar to, for example, below it is a tomato spotted wilt. You could be forgiven for thinking any of that is a fungal or a bacterial infection. It's a lesion on a leaf. You're not really going to know until you send it in for diagnostics, I guess. And we're not really going to know until we figure it out and go through all the diagnostics. So in the middle and on the right, you have a picture of sclerotinia in a lettuce field. That's just, yeah, basic wilted lettuce symptom, I guess. But on the right, you've also got a photo of that varnish spot outbreak from the lockyer. And they look kind of the same, really. Like at a certain point, I guess a lettuce drop is all going to look pretty similar. They're a bag of water, essentially. So whatever's causing them to wilt, you're going to need diagnostics to figure out, basically. Because, yeah, just looking at this picture on the right, you would kind of think, yeah, that could be sclerotinia for sure. But it wasn't. It was a lot of varnish spot. So on to control options for bacteria. your control options, let's say, are minimal, you pretty much only have copper, as I'm sure you're all fairly well aware. You know, copper mixed, however you tend to apply it. Even like that, it has pretty limited efficacy. You can get resistance from the bacteria very quickly. You also have with lettuce the additional challenge of it being able to directly contact the bacteria. Because if the infection's all hidden, you know, under hundreds of layers of leaves, it's probably not going to get anywhere near the spray. Copper also has its issues in that it needs to be in an available form, so it really matters what you're mixing it with and when it's sprayed, and like the chemistry can be quite, it needs to be quite specific in order to get the absolute best out of it. We're actually trialing a lot of different products, different chemistries, different commercial products, just to see if we can find something that might be another option for control of foliar bacterial diseases. We're currently trialing it in capsicum, but yeah, we're trying a bunch of different things with some pretty interesting results so far. So yeah, we'll keep everyone updated on that. We are looking to find some other options other than copper, basically, or an option that makes the copper work a bit better. So just to finish up, I guess, if anyone wants to send in lettuce samples, really any sample that you suspect might be bacterial, the timing and the packaging and everything is really pretty key. The thing about bacterial disease is that they're everywhere. Bacteria are literally everywhere in everything just normally. So it can be, if the sample's not in great condition, it's going to be pretty difficult to pick the actual pathogen up out of just everything else that's going on in there. Especially, as I was saying before, in terms of like a soft rot or something, anything in the soil, there's so much that's going to be there already. The pathogen might have moved on, died off, whatever, by the time it gets into the lab. So just if you can manage samples with symptoms at various stages, you know, really terrible, maybe just starting out, send us a variety of stuff as soon as you can is always great. Wrapping up the roots just to prevent anything contaminating the leaf if you're sending in leaf samples. And yeah, just a slightly damp bit of paper. Again, that's more important for leaf samples, but super dried out roots and stems don't really help either. So yeah, reasonably fresh is, I guess, what I'm saying. And just a bit about what we do with them once we get them. Basically, I will do the isolation from this lesion you can see here, that nasty black one up the top. I do the ooze test that will usually give me some indication of whether or not it's bacterial. Basically, you can see it sort of streaming out of the lesion under the microscope. We get a plate culture then that you can see underneath that. So that 1 to 2 days, 1 day is optimistic really, a couple days to make sure I've got a pathogen, a couple more days to make sure I've got a clean culture. We can also do molecular tests. That's an example of one in the gel photo right there. If we have specific molecular tests. For bacteria, you can also do some biochemical tests depending on what you're targeting. It can be quite helpful just to figure out if something's, you know, gram negative, gram positive, if it grows on these specific media or not. That can really help you narrow it down. And again, depending on what tests are available for whatever organism you're chasing, either molecular or biochemical testing can be just as fast or just as slow. That's kind of the fun of the thing, I guess. So that's pretty much it. Thank you to everyone on this slide, basically, and thank you to all of you for listening.